Good morning, afternoon, evening to all our participants from around the world. My name is Mana Shafai, and I'm the VP of the Business Development for Affinisep, and I'm the moderator here today. Thank you for joining our webinar today on exploring sample prep and instrument parameters for phosphoproteomics. Before we move forward, I would like to emphasize that this study is not funded by Affinisep, and it's done independently by ISV. The key presenter for our webinar today is Dr. Seamus Morrone of the Institute for System Biology from Dr. Robert Moritz lab. Seamus comes from a deep background in protein structural and functional characterizations. He joined Dr. Rob Moritz lab in 2019, where he has worked on the cutting edge of quantitative proteomics and deep phosphoproteomics systems biology, focusing on novel methods for the enrichment and detection of phosphosite with very low false discovery rates. These methods include automated IMAC and SH2 enrichment strategies and instrument operation optimization, all subject matter of today's talk. In summary, the key points of today's studies are the importance of phospho enrichment, how the SP cleanup method can influence phosphopeptide recovery, the instrument parameters important for phospho detections. At this time, I would like to introduce you to Dr. Florine Hallis, our product manager for bioanalysis. She will provide information on Affinisep key technologies, followed by Seamus's presentations. At the end, we have about 10 minutes for your questions. You may send them by our chat function anytime during the webinar, and we will try to answer them at the end of the session. For any unanswered question, rest assured that we will contact you directly. And also, we'll have the recorded video of this webinar published, and it will be available to you. Thank you so much again for joining us. I know it must have been difficult for some of you, and we appreciate having you here. From now, Florine will take the floor, and you can go ahead.
Thank you, Mana, for the introduction, and good morning, good afternoon, or even good evening to all of you, depending on where you are in the world. Thanks a lot for joining us for Affinisep first ever webinar on sample preparation. And before we have the pleasure to listen to Seamus' presentation on his work on phosphoproteomics, I will just give a brief introduction about the innovative SP kits developed by Affinisep for sample preparation in MS-based proteomics. So for those of you who are not familiar with Affinisep yet, we are a French company that was created over a decade ago, and we are a team of scientists, experts in polymeric chemistry or analytical chemistry. For instance, I have myself a PhD in analytical chemistry. And our main objective is to help analytical laboratories overcome their limitations and find solutions to their issues by providing technical expertise and sample preparation kits. So we are a technical company with all our activity based in France, in Normandy, with our research and development laboratories and also our production facilities. And we have also an American entity, which is represented by Mana, and a distributor network that makes it possible for us to sell our products all around the world in more than 40 countries. So like I said, we are a technical company, and we develop and manufacture all our polymeric resins from A to Z, so from the monomers to the polymers, on our unique technological platform to obtain resins that will have specific properties that best meet your needs. So as we are manufacturers of all our products, we can control every step of the process. So we can act on the particle size, on the chemistries, on the porosity to really offer you products that will answer your needs. And if you don't find what you are looking for on a catalog, or you need something very specific, you can also contact us and we can provide custom-made solution to help you reach excellent results. So we really want to innovate for you and with you. And in addition to our strong expertise in designing resins, We also apply them to sample preparation and especially to solid phase extraction, SPE. So our SPE resins are dedicated to 3 main application fields. The first one is food safety, with a wide range of molecularly imported polymers for the extraction of pesticides or toxins from various food matrices. We also have a broad range for environmental analysis, with different types of passive samplers for the extraction of contaminants from water. And last but not least, the subject of today's presentation, we have a broad range of products for bioanalysis and proteomics, and especially kits for instance, peptide desalting or peptide fractionation. So this presentation will focus really on these product ranges. So for bioanalysis, our latest innovation is a SPE membrane made of very small beads of sorbent. So those beads are smaller than what is found in usual SPE cartridges or tools on the market and that you may already have used. And these tiny beads are tightly embedded together to obtain a soft, uniform, and mechanically stable membrane with high capacity for the extraction and purification of analytes. And in addition to this high extraction capacity, our SPE membranes also present different advantages. First of all, since the membrane has self-stand, we don't need to use fritz or filters to immobilize the membrane, which greatly reduces the dead volumes and so make it possible to recover the samples with high recovery yields and excellent repeatability. Moreover, since the channeling effects are reduced with the very small distance between the beads of sorbents in the membrane, we are able to elute the compounds with a small elution volume, thus obtaining highly concentrated eluates. And if you need to perform an evaporation step before the LCMS analysis, the time required for this step is also greatly reduced. And last but not least, our SPE membranes are viable with a broad variety of sorbents, which make them compatible with pretty much all applications in bioanalysis and proteomics. So based on this SPE technology, membrane technology, we have developed 2 product ranges. The first one is called Attract SPE Disc. So this is a comprehensive range made of polymeric and silica-based sorbents. So it can be C18, HLB, or GVB, for instance. So very chemistry oriented. And the second range is called BioSPE, and this one is more application oriented because it's composed of ready-to-use kits, specially developed and optimized for different applications such as peptide desalting, peptide fractionation, glycopeptide enrichment, or purification of intact proteins. So based on the SPE membrane technology, we have developed 4 different formats for microelution and microextraction when you work with small elution volumes, which is often the case in proteomics. The first format is the stage tips. So that's we also call SPE tips. We also have developed the spin columns, the SPE well plates, and the SPE cartridges. And all these different formats are also available with different sizes. For instance, regarding the SPE tips, we have 10 microliter, 200 microliter, and 1 milliliter tips. For the spin columns, we have 4 different sizes, micro and mini spin columns when you work with small volumes, and 15 or 50 milliliter spin columns when you work with higher volumes. For the SPE well plates, we have 2 types of 96 well plates. The microelution plate, for which the tips have the same shape as a stage tip, the wells have the same shape as a stage tip, and the classical 1 milliliter plate. And we have also recently developed a 384 SPE well plate microelution for a very high throughput experiment. And these plates also have wells that have the same shape as a tip. And finally, for the SPE cartridges, we have 1 milliliter, 3 milliliter, and 6 milliliter cartridges available. So we cover a wide broad of formats. And in addition to this flexibility of formats, we also offer a real flexibility of capacities because all our formats are available with different binding capacities. That is to say, different thicknesses of the SPE membrane to best meet all your needs. So all our tools offer a real flexibility in both thermal formats and capacities, and they are you can use them with sample volumes ranging from few nanoliter to several milliliter, with protein or peptide amounts ranging from few nanograms, so single cell proteomics, and up to several milligrams. So now we will focus more on the characteristic of our different tools, starting with the spin columns that are very useful for high protein or peptide amounts, as they have high capacity of extraction. And these spin columns are very easy to use because they are simply used by centrifugation. And since they have a lid, they can be closed, which makes it possible to perform incubation directly inside the spin columns, or even store sample before LC-MS analysis. Regarding the 96 or 384 well plates, so they are used for high throughput experiments, and they can be partially used. You just need to cover the unused wells and use them later without any consequences on the results. And you can also use them with different methods, either by centrifugation, by positive pressure, or with a vacuum manifold. And finally, for SP cartridges, they are used to concentrate large sample volumes in small elution volumes, and they can also be used either by positive pressure or with a vacuum manifold, and they are compliant with pretty much all SP automates available on the market. And as we use the same SP membrane technology for all formats, it makes it very easy to change the format or scale up the process if needed. Now, regarding our SP tips, they are packed using fully automated methods. So if some of you have already done their own stage tips by cutting some disc and put it inside a pipette tip, you know that it takes time, that is pretty annoying. So our tips helps you save time and stop wasting time by providing already packed tips with no tip-to-tip variability and highly repeatable process. So you can let our automate, that we kindly nickname Jack, do all the job for you, and you can use your precious time doing anything else but preparing your own stage tips. Another aspect important about our tips is that, contrary to other tips that you may have used that are used by pipetting, our tips were conceived and developed for an optimal use by centrifugation, which is less tedious and painful than manual pipetting. So you can say goodbye to thumb tendinitis due to repetitive aspiration and dispense. And centrifugation is also more repeatable than pipetting, as we have shown in one of our application notes. And once again, you can save time by centrifuging several samples at the same time. And as we really want to simplify the use of our products for you, we also supply kits that will contain DSP tips, of course, but also spin adapters and collection tubes. So you just have to centrifuge the tips, and then you collect your purified samples at the bottom of the tube. And so it's very easy and very fast process. So our tips can be used by centrifugation, like we just saw, and we have a video on our website that explains how to use the tips by centrifugation for peptide desalting with a step-by-step protocol. But our tips can also be used by positive pressure. And in this case, you just have to load the solution from the top of the tip, and then you apply pressure to make the solution go through the sorbent, and you recover your purified sample. And this method of use makes our tip compatible with most sample preparation automates for fully automated workflows, like for instance, with the Digest Pro, the Resolve X800, or the Evosep one. And so talking about automated workflow, here are some results obtained during a work that we've done in collaboration with CEM and the P3S platform from Sorbonne University on the development of an automated method for desalting of peptide on the Digest Pro, and comparison between our Attract SP disc tips C18 and homemade stage tips with the desalting of 100 nanogram of Eli Digest. And you can see here that on protein identification, desalting with the Attract SP disc tip C18 leads to 9% increase in the number of protein identified compared to homemade stage tips. And also, if we look at the polarity of the peptides retained, we can see that with the Attract SP disc tip C18, we retain more the hydrophilic peptides, so we offer a wider spectrum of interaction compared to the homemade stage tips. On a quantitative aspect now, the study showed that with the Attract SP disc tip C18, you can get a 25% increase in the peptide intensities compared to homemade stage tips. And looking at the clustering, it shows that we have two distinct groups for the homemade tips and the Attract SP tips, with less missing values for the Attract SP disc tip C18. So this work was done for automated desalting, but like I said, our tips can also be used in manual studies or by centrifugation. And here are some results obtained with the Institut Curie in Paris, who compared our different capacities of our tips, so T1 and T2, T2 being twice the capacity of T1, and they compared our tips with the competitor C18 tips for the desalting of ELI digest by centrifugation. And the results showed that with our tips, we are able to identify more than 97% of the proteins present in the digest before desalting, with a good retention of peptides on a wide polarity range, thus enabling to identify more proteins than the competitor tip, and also offering very good repeatability with low variability observed, thus highlighting the reliability of the Attract SP disc tips. So these tips can be used for desalting of native peptides, but also for TMT labeled peptides. And spoiler alert, it also worked very well for phosphopeptides, but I will let Seamus talk more in details about that in his presentation. So that was just a very quick overview of the different products that we offer. And if you want to have more information, I invite you to go on our website where you can find more information about our different products and application with different booklets and brochures. And we also have several posters on peptide desalting or peptide fractionation available. And of course, if you have any question, don't hesitate to ask them now. We can answer them at the end of the webinar. Or if you have question or request in the future, you can also contact me by email here. Thanks a lot for your attention.
Thank you, Florine, for that great presentation. As she said, if you have any questions, please feel free. You can send them through chat here to us. Please just put her name first. If you have any questions about the products and pricing and availability, you may address them to me as well, and we go from there. Seamus, I think now you have the floor.
Thank you, Mana, for the nice introduction, and thank you, both you and Florine, for inviting me to give this talk. So let me share my screen. Okay, so I'm Seamus Morrone. I'm going to talk today about some phosphoproteomics work we've been doing in the Moritz lab regarding sample preparation and instrumentation on LCMS, specifically on Orbitrap for DDA. And as Mana mentioned, I just want to make clear that this was all carried out at ISB without any sponsorship or funding from Affinisep, because later on it'll seem that I'm promoting them, but this was all independent of any interaction with them. So since we're all attending this webinar, I'm sure we're well aware of the importance of protein phosphorylation. It's arguably one of the most important post-translational modifications that proteins undergo in all domains of life. It's highly dynamic and reversible, so it can act as a number of different roles in the cell, such as in state transitions, molecular signals and switches, and timers. And this is highlighted by the role it plays in one of the central processes of eukaryotic cells, the cell cycle pathway. Yeah.
Sorry for the introduction. We cannot see your slides.
Oh.
We're not able to see your slides.
Yeah. So I don't know if there is a problem with your screen sharing, but
Try again.
yeah,
Okay.
sorry for interrupting you, but
How's this? Can you see it now?
it's still, yeah, now
It's loading.
you see it.
Okay. All right.
Thank you.
Yeah, sorry about that. So cyclin-dependent kinase undergoes phosphorylation in order to be active, and as its name implies, it in turn phosphorylates other proteins. So you can see phosphorylation plays a large number of roles just in this pathway. It's also estimated by researchers that up to 75% of the human proteome may be phosphorylated, and this is at one time point or another, or in one tissue or cell type or another. And so this qualification I made is because it's highly dynamic and it can change very quickly within short time frames or short distances. And as you can imagine, because it plays such a central role in so many different pathways, that when it goes wrong or has a dysfunction, that it could lead to a number of different disease states. And particularly, it plays a large role in multiple forms of cancer, such as when the receptor tyrosine kinase signaling pathways go awry. So not just of interest for its general biology, but also for any potential therapeutics it may play. So there are a number of different ways of detecting phosphoproteins in your samples, but I would argue that the gold standard is still mass spectrometry. And this is for a few reasons. It provides good sensitivity. It can cover several orders of magnitude of dynamic range. So you can capture not only abundant proteins in your sample, but more rare and less abundant proteins. It's got very high throughput, capable of analyzing hundreds of samples per day. This is an ongoing field that keeps pushing this further and further. And not only can you run samples subsequently, but you can also multiplex by combining multiple samples into one master sample and run them in parallel by using labeling. And not only does it provide excellent identification and quantification, but it also provides useful information on the localization of the phosphorylation. This is important because most phosphoproteins have multiple potential sites as well as multiple states of each one of those sites, and that will alter its biology that it plays in the role. And besides all these, there's also a well-developed field and background with several publications on sample preparation and instrumentation, going from your sample sources, either cell types or tissues, all the way through protein extraction and digestion, and then the subsequent enrichment steps with multiple different methods of capturing the phosphopeptides, and then on to the instrumentation LC-MS portion. But as with anything that's worth pursuing, there are a number of challenges of phosphoproteomics. The main one, which I have talked about briefly previously, is its dynamic nature, so that a sample processed at one time point may be very different from the exact same sample processed at a different time point. Because it's also present in low stoichiometry and low amounts, that you require generally more moderate amounts of starting material than if you're doing something like a standard lysate digest. And because of this stoichiomic nature, that it's in low abundance, it also requires additional processing steps than typical preparations in order to get rid of any non-phosphorylate components, and this is the enrichment process. So I want to cover each one of these points very briefly. So on to this first point about phosphorylation being dynamic. This is illustrated by these two figures from these great publications, been around the field for a while. That samples processed at one time point may be highly divergent from same sample. So on the left hand, we have a tissue sample that has been excised and just left on the bench for the time indicated, up to 3 hours, and then blotted for the presence of AKT and a phosphoform of the AKT. And what we can see is while the total AKT remains relatively constant over this time point, the phosphoform is rapidly lost, even within 30 minutes. It's down to low levels. So this indicates that timely processing of your sample is going to be important if you want to capture these phosphostates. On the right-hand side is an illustration of the complex dynamic nature of phosphorylation, where a sample is stimulated by EGF, and then its samples are taken up to 20 minutes post this stimulation. And as you can see, even within 5 minutes, the changes in phosphostates, both of different proteins as well as in the same protein at different sites, changes rapidly and changes very heterogeneously depending on what site it is. So you can imagine if you take a sample at one time point versus another, you're going to get very different results. Onto its low stoichiometry. As the field is progressing rapidly towards smaller and smaller sample amounts down to single cell analysis, phosphoanalysis still requires moderate amounts of starting material. So while we're dealing with sub-nanogram amounts in a standard lysate digest, we're still working with microgram amounts up to several milligrams of starting material for certain phosphoanalysis, such as phosphotyrosine analysis. And so it's a precious sample that you're using. You want to make sure that you can get the best out of it. And also tied up with this is that it requires additional processing steps. So we can illustrate this here with an unenriched sample run on LCMS system and then analyzed, and we get back a certain number of spectra, peptides, and proteins identified from this sample. But of these, only a very small subset are actually the phosphoforms. So they're present in less than 1% in the spectra and peptide level. Conversely, if we run an enriched sample, while we're getting slightly lower number of spectra and peptides and proteins, the vast majority of these are phosphoforms. And so enrichment is necessary if we want to get any meaningful data with some statistical power behind it. So in order to get the best out of your sample, you want to have a good phosphopeptide processing pipeline in place. So you can kind of divide this into two sections, the sample prep section and then the analysis section. And I'm going to spend most of the time on the sample prep section for this talk, but I can touch briefly on the analysis method. I just want to take a quick moment to plug the transproteomic pipeline because this is a software suite where you can go from the raw file generated by your mass spec and to a final tabulated and quantified form at either the peptide or protein level, whatever is of interest to you. And it incorporates a number of different identification softwares and validation levels and quantification levels. And it's free and open source, developed at ISB. It's constantly updated. There's a new version coming out, 7.1.0 very soon. And not only this, but there's a strong community participation in it. There's an online discussion board, and you're always free to email any of the developers with more questions. So I would recommend it, of course. So the first part, this processing method pipeline, we're going to need several features of this in order to generate good data. So we want something that's robust and reproducible. Usually that means less steps and less manual intervention to limit errors and variation. And then we want faster processing time, as we saw. That's timeliness is important. So in our lab, we've been focusing for the last several years on protein aggregation capture using magnetic beads. And this is based on the work of a number of excellent labs that have been developing this and have shown that it's very useful for sample prep, not just for phospho prep. And it's a fairly simple and straightforward method. You start with your proteins, you aggregate them by the addition of organic solvent. You add in your microparticles that then the aggregates absorb onto them. And you can pass them through multiple washing steps to get rid of any of the chaotropic agents and salts that you started with, and then buffer exchange them into an appropriate digestion buffer. We usually choose something that is volatile, such as TAB or ambic. And then you perform the digestion. You've got your sample ready for either drying down or proceeding with the next steps. And previous work has shown that this is superior to general in solution method for phosphoproteomics. And it's also easily incorporated into automation by using robots such as the Kingfisher, because we are working with magnetic beads. It can easily be manipulated with a robot. And it's also easily incorporated into a phosphoproteomics workflow. So the second part is the phosphopeptide enrichment. And we have been using mag resin beads from Resin, the company, because we've found that these are very easy to work with, and they yield very good results. Previous publication showed that the zirconium and titanium iMac versions can capture a wide variety of phosphopeptides with a lot of overlap, but also they capture partially unique subsets. So what we do is a two-pass method where we enrich first with the titanium iMac, and then we enrich with the zirconium iMac on the same lysate sample, same digest sample, and then combine these two elutions into one. And we use the high throughput beads, the HP beads, because we have found in our hands that we have a better recovery of phosphopeptides with the HP versus the standard iMac that they provide, but they provide both of these. And this is also all done on the Kingfisher, so it's easily automated and highly reproducible. And the third part is usually the cleanup after the elution and then drying down for mass spec. And we've found that the choice of your SPE, the solid phase extraction method, matters quite a bit for your sample recovery. There's many options available, as Florian was talking about. There's cartridge formats, tip formats, plate formats, and they range in the volumes that you can apply and the capacity of the matrix and the elution volumes. And as I said, we've found that depending on the choice, we get better or worse recovery of phosphopeptides. And so for the last few years, we've been using Affinisep's Attract SPE disc tips. And as Florian was mentioning, these are pretty fast sample prep. They can be used in microcentrifuges or tabletop centrifuges. They're easily scalable up to 96 well format. You can just stick them in a plate and centrifuge those. They come in a wide variety of different formats, from the 10 microliter, 200 up to 1000, and a 96 well plate. And they have varying capacities from the low microgram range for the 10 microliter format up to over 100 micrograms for the 1 mil format. And it's a very quick, basically 6-step prep. You just condition and equilibrate the tip, apply the sample, wash, and then elute in a small volume, which is very convenient for drying down, or if you can directly apply it to your LCMS system. And so we found that when we compared it to several other brands and formats, that we were getting superior recovery of phosphopeptides using the Affinisep tips. And in this, we're illustrating it with EvoTip, that we were getting a better recovery. Even though we were using the recommended protocols for all of these, the recovery for Affinisep was much better, although we have been using the EvoTips quite a bit for other sample preps, and we were getting very good recovery. For some reason, we were getting worse recovery with this. And we were kind of wondering what it was that was driving this difference, and we were suspecting it was our elution conditions. And what we found was that there is a difference in the populations of phosphopeptides that were being captured by Affinisep versus the EvoTip and other brands. So we found a slight shift in the gravy score for the EvoTip in this instance towards more hydrophobic peptides being captured. And this was also reflected in a shift in retention time towards later retention times, and a loss of the small M over Z value precursors. So this was indicating that the Affinisep tips were capturing most of these peptides, whereas some of these smaller, more hydrophilic peptides were not being captured by the evotip in this instance. But when we looked at the reproducibility within the 3 replicates of all these, the evotips and the Affinisep tips all performed very well with high reproducibility. It was just that, and this was about 60 to 70% of unique phosphopeptides, so not just strip peptides, but the actual phosphosite being incorporated. And it was, of course, even higher when we looked at the best 2 out of 3 for all of these. They were very similar. It was just that the Affinisep tips seem to be capturing more phosphopeptides. This was also reflected in the different formats of Affinisep tips. So they captured most of the phosphopeptides that the evotip were capturing, but each one captured slightly different subsets of these phosphopeptides. So while there was a large overlap, there was still a number of unique ones that were being captured by the different T1, T2, and T3 formats of the 200 microliter. But it was still a very high reproducibility, even when we looked at the overlap of the 3 different formats of 200 microliter. It was very similar to within the format replicates, 60 to 70%. So this illustrates that these Affinisep tracked SPE discs were very robust and very reproducible. So this was one reason why we were focusing on using these. And it wasn't just the evotips that we were comparing to. So we did compare to different formats. We looked at one cartridge chip, format, the Atlas column, as well as using a zip tip, and performed the cleanup using the recommended protocols. And we still found that the Affinisep had the best recovery of all of these. And the Atlas column, the cartridge format, had the worst. And this may be because it was using a completely different cleanup system, and the beads may not be as fine as on the tip formats. And it also generally is a larger volume that you're working with the Atlas column. And the scalability for the tip format is generally better than the cartridge format. So we prefer using the tip format over this, as it's also a lower elution volume. But we also wanted to know if we could use the same protocol for a different tip. So if we switched, I mean, mainly there's the differences in the protocols deal with what acids to pair your organics and washes with. So we wondered if we could switch these acids around, and would it affect performance? Maybe this is one reason why we're getting these differences. And so if we switch the Affinisep tips from acetic acid to formic acid, we actually didn't find much of a difference in recovery of phosphopeptides. They're all very similar at both the non-localized as well as the localized level. But we did find a slightly better recovery using acetic acid with the evotip. But in both instances, the Attract SP Affinisep tips were performing better, and this was more pronounced at the localized level. And then one final thing that we investigated was during the quenching of the enrichment. So after enrichment, when you elute in a high pH condition. And so what we generally do is acidify this immediately afterwards with TFA to about 5% in order to bring the pH down to 2, and apply that to the cleanup column. And this was one reason why we were suspecting we were getting these differences, that the Affinisep tips can just withstand this higher acid better than others. But there was a paper that came out about a generation ago that showed that the addition of phosphoric acid during cleanup on a C18 column with phosphopeptides improves phosphopeptide recovery because it acts as a passivating agent so that phosphopeptides aren't irreversibly bound to the matrix. So we wondered if we could just replace the TFA with phosphoric acid instead. So we tested several different percentages of TFA as well as 2, 3, and 4% of phosphoric acid. And what we found was in all the conditions tested with the 2 different acids, the phosphoric acid had better recovery, although it wasn't significant except at 3%, where it was quite different. And so we focused on this 3% for later on, but we are looking at the differences in the populations of phosphopeptides within these different samples. And we found that for lower acid percentages, we were generally getting more single phosphorylations. And for higher percentage for TFA, we got more hydrophilic and smaller precursors. And the phosphoric acid, it was similar, but it wasn't nearly as pronounced as the TFA conditions. So this shows that certain percentages of acids, you may actually be losing some of your subpopulations of phosphopeptides. So just as a quick summary, most of these tip formats are, small elution volumes, which is a plus, and very adaptable format. Overall, we found that the Affinisep tips are very robust in changes of protocol, different acids being used, and very highly reproducible. And the Atlas column is probably going to be best for larger samples, as the formats we were using can have a capacity up to several hundred micrograms. So that might be best when you're using large samples and large volumes. So I just want to touch briefly on this final portion about the analysis methods. I'm not going to have much time to get in too much detail, but there's other talks, and I've actually given a talk about this before, so you can email me if you want to link to that. But generally, if you're using your Orbitrap and you generate a method, you usually get auto-filled values for your parameters that are of interest. And so if you just use these out-of-the-box settings, it's actually optimized very well for your unenriched samples. So on the left here, we have one of these out-of-the-box settings, the standard method. And then on the right is a method that has been optimized for phosphodetection, as well as just lower post-translational modification abundance detection. And there's similar amounts of unique peptides that were identified between these two methods. But if we look at a phospho-enriched sample, we get a boost in terms of the unique phosphopeptides at the unlocalized level, but this is really brought out in the high confidence localization, where we're almost getting half again as many identifications as in the standard method. So it's important to have your method optimized for phospho detection. But the question is, there's so many parameters, which ones are the important ones to optimize and which are the ones that generally you don't need to worry about? And you have at the MS1 and MS2 level for Orbitrap instruments, you have maximum injection time and automatic gain control, which kind of have a dependent relationship to each other. You've got the collision energy used, you've got the number of precursor selections, you've got the gradient length, and many others. So which of these should be optimized? And my take-home message is that it depends on what you're after. Do you want high sample throughput to get through as many samples per day? Do you want just high phosphopeptide numbers without worrying about their localization? Or do you want high confidence in those localizations at the sacrifice of less identifications? And there's lots of good studies on Orbitrap instrument parameter optimization. I would recommend looking into these papers that have covered a wide variety of different sample types, including even phosphopeptide samples. But we have also done some work on this, and we have a draft paper on Chem Archives that you can look into if you want more details on this. This slide kind of summarizes a few of the values we looked into, and you can go over it, but it's basically that there is always a trade-off in choosing a low value or a high value for these parameters, the AGC and IT at the MS1 and MS2 level. And so, the take-home message is, what do you want from your experiment? Do you want fast processing time, localization, large numbers? So that will really depend. That will dictate what you want to input into your parameters. But probably most important, even more important than what parameters you're choosing for your LCMS, is your sample preparation. it's pretty much will dictate what your results will look like in the end, because the old adage, garbage in, garbage out. So you want to make sure you got your sample prep down very nicely. And optimization, as I mentioned, it involves trade-offs. You're always have to sacrifice one thing for the other. So you're not going to get a great method that's going to have high numbers and high localization and high throughput. So you're going to have to pick and choose. And of course, there's many other variables that I have not touched on that are touched on in our paper and other papers, and some that we have yet to talk about in detail. And so I just want to thank Rob Moritz and the Moritz lab, everyone in there that has given me help over the years and assisted me. And so now I will take any questions.
Great. At this time, I guess we only have a few minutes left. I wanted to thank everybody for attending this webinar. I hope that you enjoyed and benefited from this study. We wish you a great week ahead, and please reach out to us with any questions. Have a wonderful week.